I'm Rob Burgess, and I head up Worldwide Business Development for Sino Biological. I just want to remind the research community that, among other products, we have the world's largest selection of full-length recombinant proteins made in mammalian cells. In addition, we have a very comprehensive line of products related to coronaviruses, and specifically the recent COVID-19 outbreak. These include spike and end proteins and receptors, antibodies, and even testing kits. For more information, please check out our website at www.sinobiological.com. Thanks for your time.
Okay, thank you everyone. Welcome to the webinar this afternoon. I'm delighted to introduce our panelists for this afternoon. We have got Pascal Brennesstein, who's an advisory board on BioCopy, Dr. Gunther Proll, he's a BioCopy senior scientist, and Gunther Roth, he's the CEO of BioCopy. I'd like to hand you over to them. If you have any questions, do enter them into the chat, and we'll be dealing with them at the end. So as it is, I'll now pass you over to the panelists. Thank you very much.
Okay, I hope everyone can see me now, and or at least my monitor. And as introduced, I'm Gunther Roth, CEO of BioCopy and founder. Many thanks for the presentation today and for your attendance. And I hand over to Pascal, as he may introduce as the most business-driven man here in our group. To introduce BioCopy, and then we will take over from time to time. So, Pascal.
Thank you very much. If you could switch the slide, yes. So I think quickly on the summary, how we're going to go through today. I mean, BioCopy. I think we as a company, we copy your DNA, we copy your RNA, and we copy the proteins, right? So, you know, that's the core essence and the core technology that BioCopy actually has. So I'm going to talk quickly about some of the core historical elements of copying technology. Then afterwards, we will talk about how to copy and paste biomolecules. That's Gunther Proll, see the label-free virus scores. And then we have Gunther as well to talk about some of the collaborations and tomorrow's universal vaccines moves. I think it's the right time at the moment, given COVID, to really talk about how to speed up some of the vaccination processes. So BioCopy has a board, and I'm part of the board. So please, Gunther, if you can take the next one. So I mean, you see here the combination of the team and the board. I think on one side you have on the left side with Gunther, a person, a scientist, as really from the bottom up, I mean, biochemist, physicist, microsystem technologist, and screening and assay expert, as well as a adjuvant developer for vaccines. You know, many years back, he is actually he's the inventor and the founder of BioCopy. He actually at the same time has been co-founding some other companies in the past, and he worked jointly already between 2004 and 2008. And published together with a company that you all know, CureVac. So I think here are some historic back scientific activities that Gunther was already pushing here, right? Beyond that one, he had a joint project 2018 with Update, which was part of BioNTech. Now, the other scientists in the team, Gunther Proll, founder of Biometrics, at the same time here the entrepreneurial spirit, inventor of score technology, and again another expert in assay development and analytics. If you then look on the left bottom right side, you have Alexander von Gawijn. He clearly is a first generation developer of vaccines. I think, for example, Japanese encephalitis, where actually he was co-developing. He is still applied nowadays, as you all know. On top, he's founder of Intercell, which became Valneva, which is a vaccine company with about 3-digit million European euro turnover. Then on the commercial side, you see Rainer Böhm and myself, we come out of Novartis. I think Rainer having there an expertise as former Chief Commercial Officer at the Pharma Division and CEO of Novartis of Pharma. He managed as well the marketing and was head of international marketing in Novartis. And myself, I was as well 24 years in Novartis, and then made the different moves into biotechnology as well, including a regional responsibility in Shire for Eurasia, Middle East, Africa. And always for the last 6-7 years, I've been coaching startups with a focus on pharma, digital high tech, and digital biotech. So you see here a team, an international team, bringing all the assets which are important to make best leverage and get that technology of BioCopy to the next generation and bring it to market. If you go to the next slide, please. So what are we talking about, right? We're talking about microarrays who enable high throughput binding, but on the other side, the production is really high tech, it is expensive, and you have some challenges there, right? So microarrays are basically small dots in a small space, to say very basics for a non-scientist like me, enabling hundreds and thousands or even millions of biochemical measurements in one single experiment. So even today, microarrays are the core technology used in the next generation sequencing, like for example in Lumina. But at the moment, today's microarray production is sort of at the dead end, right? So I think here you can see two opportunities. Here you have on one side off-chip, where you can put production off-chip. So you have to go through synthesis of each amino acid of each pair bit, and bit by bit you have to move that forward into molecule, and that's a tremendous effort, time, and cost assuming. And as you all know, due to its synthesis, it's pretty restricted, and purity and the yields that you're trying to achieve is sometimes hard work to even get it there, right? And then on top of that, looking from off-chip, go down to on-chip. So on-chip, so if you're looking into the on-chip situation, that you have the yields for on-chip is clearly, I think, improved. On the other side, the biosynthesis of the molecules in cells, purifying them and transfer them into a chip is again a challenge. So this usually yields best quality and theoretically unlimited molecule length, but it takes time. So about 100,000 compound at one single second transfer time is more than a day per one single array to print. So if there's a technology to speed up that process, that actually would be very helpful, right? So why not going back to the Middle Ages? When nowadays, of course, the digital technology, digital century nowadays, but there used to be 30 years ago, 40 years ago, we had traditional books. And maybe you remember going back to the Gutenberg technology at the time to basically print books, coming away from individual books being handwritten to printing them. And at the end of the day, the latest technology then beginning of the 20th century was the Xerox copy machine. I guess we all somewhere do have still some Xeroxes, right, or some printing companies, printing machines. But still, I think what would be the possibility if we do the same for biotech, but the biochemical world here, right? So if we consider now biomolecules, for example, as words or as a sentence, and made out of a building block, a letter, and then we think about an amplification of books, as I was mentioning before, and enlarge that part, right? So looking into microplates, which we have here for biological assays, which are dating back to the mid-50s. And from these microplates, then having some 100 data points in there, but you know, we still are nowhere here on giving the latest technology we have on some of the genome sequencing we have here. So to microarray with the respective limitations, there should be actually new technology. And what about having here a step up like a Xerox, a DNA, an RNA, a protein copying technology, which will get us to a next level of having speedy experiments, speedy replications with great output. And let's see, I think we have here something on BioCopy that we would like to introduce to you. So I pass on to Günther. So thank you very much.
Thank you too.
Oh, thank you, Pascal, for this inspiring introduction. So, yeah, well, oops, moving. Okay, well, because copying is our technological core, I first want to share this photo of the world's first Xerox with you. So this Xerox was created almost 80 years ago in New York City, and now what we see here is our master, so to speak, our DNA master comparable to a masterpiece of paper. It consists of labels, DNA molecules in a microarray format, of course. You also see the scaling here compared to the original Xerox. And well, we use this DNA master to make copies from it. We copy DNA to DNA on a microarray, which was published quite some time ago. But we also copy from this DNA master proteins. So we also have the possibility to create protein arrays from the DNA information. So we at BioCopy really can say that we have invented something like a biomolecule copier, which allows us to copy almost every DNA template into DNA, RNA, or protein. Our proprietary technology, of course, is written down in several patents. 13 of them have been granted so far. We have 2 more pending, and more than 50 applications discovered based on our technology, which we are using and moving forward within our company. But the most important then, I want to share with you today, as shown here. So first of all, we are using this technology to identify new vaccine precursors within very short time. So almost 2 days here to have something enhanced to start with a vaccine development. We are performing mutant profiling of current vaccines, and a very interesting topic, of course, is the search for a universal vaccine, which means we want to see possibilities to develop vaccines for mutants which are not there at the moment, but which will come up in the future. And of course, we also use the technology to develop new diagnostic applications, especially here the test for the immune status of patients after vaccinations or after infections. To start a copying process, first of all, we use a virus, for example. And what we extract from the virus is, of course, its genetic material, and this is then cut into pieces. And we use then these pieces of genomic material from the virus to create our master DNA slide, so to speak. And with this DNA masterpiece, we can create a protein microarray, which means we have now all these informations translated from a DNA strand into a functional and folded protein on the surface of a glass microarray. What can we do now with these protein microarrays? What we perform at BioCopy is an assay where we use the blood or the serum samples of survivors, and to perform assays where we see the binding of antibodies out of these samples to the individual proteins on this protein microarray. And the hits we see during this binding assay is the starting point of new vaccine. These are our vaccine precursors we are aiming for, we are hunting for at BioCopy. Well, but how can we detect the best hits from this assay? I think that's also one more important story at BioCopy. At BioCopy, we use a technology, a readout technology, which we call SCORE. SCORE stands for single color reflectometry. And as a very simple introduction, I like to explain it in a way that it's very similar to the situation we have with soap bubbles. Soap bubbles are nice, colorful things, and the color comes from the interference of white light at the soap bubble wall. Depending on the thickness, the local thickness of the soap bubble wall, we have different interference conditions, and therefore we see different colors from a soap bubble. And our idea was to transfer this basic interference idea into a sensing setup, and we implemented a label-free detection technology based on this idea, which offers the possibility to investigate this interaction of antibodies with proteins in real time in an imaging mode, and really also in a quantitative mode and manner. Just to give you some background about this technology, I want to briefly introduce the physical, the physics which is behind this technology. Everything is based on white light reflectometry. We worked with these technologies many years ago in a way that we illuminate transparent transducer from the backside with white light, and beside the transmission of the white light through this transparent transducer, we also have reflected light beams. Reflected light beams from each interface. You see here one from the glass interface to the biolayer, which is here in blue color, and the second one from the interface biolayer to the buffer, which is just above this system. And these two light beams can interfere with each other on the way back to the detector. And what happens now when a biomolecule is binding to this layer, we see that the thickness increases, the refractive index changes over time. We see over time a shift of this reflectance spectrum because it's white light, and we can see the association over time, the dissociation, as well as the regeneration. So, well, this is complex technology. I think maybe you have heard about biolayer interferometry, which is also on the market, which uses this technology, this principle of sensing, but we developed it to something new. We thought about this experiment once more, and what we see is that when we just look to the right wavelength, we can see and get the same information out of the same experiment, but we don't need to measure the full spectrum. We just look at one specific color. So this change of the technology is the reason why we call it single color reflectometry, and it transports the same information than the white light basic technology. And it's really working. What you see here is a movie taken with a camera which really sees these interference intensity changes on a microarray surface. We see here the binding of an antibody to 500 different spots of a microarray on a surface in false colors, of course, because it's label-free, it's no fluorescence, and because it's time resolved, we can extract from this movie for each individual spot. You see here a binding curve, a sensorgram. We see the association phase, we see the dissociation phase, and for some experiments, we also can regenerate surfaces. And the nice thing about this technology is that we can extract also kinetics informations. We can see association rate constants, dissociation rate constants, and the thermodynamics constant, the affinity of the interactions. So I think this is the perfect match of a detection technology to the microarray world. So what do we have at BioCopy? We have really a smart technology to produce microarrays in any kind in terms of biomolecules. This can be DNA, RNA, protein microarrays, and we have combined it with a very unique label-free real-time detection technology. So with this technology combination, we can run assays up to 20,000 interactions in one shot to investigate biomolecular interaction, really for complex samples like serum samples or whole blood samples. So with this, I hand over to Günther, who will present you more nice results we have produced with these technologies.
Yeah, many thanks. I have the pleasure to present you the colorful images instead of the hard facts for the physics behind that. But always think of the soap bubble, then you know, okay, it works basically, and it's really colorful at the end in the readout. And I like to give you some impression what we do at BioCopy before we start with the COVID-19 part. So basically, we have a lot of applications where we can have, for example, if we have copied DNA as single strand, we can use them as aptamers, and we can also synthesize them with according partners, make the readouts. You see here several hundreds of readouts in one go with 1 to 10 microgram of material. You get these results. If we copy, we are not limited in resolution. Basically, you can see here how thin and fine the resolution can be. It's even so low that the microarrays cannot get problems. And on the other side, we can work with peptides, for example, as well with whole proteins. And as you can see here, as the copy step is not limited to some round and rectangular structures, we can even do hexagons or even any fancy structure which can simply be filled with water at the end. And you see here also a multiplex assay where we bound several different targets to the surface and showed that they have their specificity and their interaction with different other setups. And this enables us to really go into a deepness of analysis which is not possible so easily with a Biacore. You can do that also with a Biacore, no problem, but you can do only 1, 2, 3, or 5 lines, for example, but you can't do it several hundreds in a go. And you can do the same measurements with a BLI, but if you imagine to make 1,000 BLI measurements, ask your scientists in the lab how long they will take for that. And now let's switch to the COVID-19 assays and also make the bridge to Sino Biologicals for the cooperation. With our screening system and the detection system Günther introduced, we can basically make a pseudo-neutralization mutant screening. So think about, we take the original Wuhan type, we take a lot of mutations, bring them on a surface, either copied or printed, and then you have already a high number of different mutants on the surface, and then you basically flush the serum of a person over it, either vaccinated or pre-infected, and then you simply measure the binding curves. And then in a second step, you measure, does the ACE2 bind still against these S proteins, for example. If it binds, that means that you're not protected. If it doesn't bind, it means you're protected, basically. And we did that in a complexer assay. And to bring you all to the same knowledge here, I basically give you the sensorgram, how it should look like, and 3 different comic molecules, so to say. So let's assume we make a blocking. There should be nothing in the measurement. And then if we flush serum over the surface, we should get some reactions. If the antibodies bind, we get a high reaction. Typically, also serum makes some unspecific interactions with any molecule. So also in the red molecule, you get a little bit of a signal. We make an intensive washing step, and after that, we go with ACE2 over the surface. And if the molecules are blocked, we get no signal, so the green line stays low. Whilst if the ACE2 binds, the signal goes up for the red and the yellow molecule. So this means here are proteins, antibodies binding, but they don't prevent the binding at the end. And basically, this is all what we need. But as some core scientists still like to have some fluorescence, we can basically put some fluorescence marker at the end. So we have secondary antibodies just as control at the moment and to validate our technology. We flush an anti-human antibody with a green fluorophore and an anti-ACE2 antibody with a red fluorophore over it. And this combination gives you a very fine traffic light-like color map. So basically, you can say red is bad, green is good, yellow is we don't know. Maybe it helps, maybe not. And this enables you at the end to look at the fluorescence, but as said, basically for the label-free detection, you need only the first part. And for the transmission, if you, it's a little problematic to bring all the movies in the internet. If you're interested, contact us. We can give you then confidential information after signing a CDA. But now you can see the official parts. So basically think of that we made an array with 90 mutants. This is only a simplification here for the moment. You will see the arrays in the next slides. Basically, we have some controls. We have all the other coronavirus on the surface, the S and the N proteins as also control and see how the people are pre-infected. And then we have an area where we have a lot of S and RBD mutants. And then basically you can use therapeutic or the diagnostic antibodies like shown here. So basically, if the diagnostic antibody works, everything is green and fine. If the diagnostic antibody doesn't work, you simply miss some green spots because it doesn't detect or bind to the molecule. And if it's a protection assay, basically, then here it's very, very important that you don't have any red, because red means that your therapeutic antibody will fail. And in the last months we had several great companies which had to say, okay, our protective antibody fails against this mutant, that mutant, and maybe also this one. We can measure that, and we see that for some of these antibodies already way last year. If you look for infected persons, for example, you can see on this area if there had been some pre-infections with some other coronavirus, but you see typically no protection against any of the S proteins to our knowledge. And directly after infection, you see that there is some good protections up against some of the coronavirus mutants, but not at all, for example. And very prominent, you see it also later in the data, is the E484K mutant, which is a core mutant for UK and Brazil, as well as South Africa, and also in some India and I think also California it comes up. So we see that already in that data, and I present that in a larger part in a minute or two. You can use that basically also with a vaccinated person. This gives you then an impression. Does the vaccine cover all the mutants? How good and how fast does it cover it? So it could be some kind of assistant measurement for phase 1 to 3, but also for phase 4 for the monitoring if new mutants come up. But even in the preclinical phases, you can look for formulations which may cover better different mutants in that case. So the array was made in that case purely by printing, because we realized here our copying development is at the brink to realize here that, but not good enough for all the mutants, and also gaining all the mutant DNA is really difficult. So we had a talk with Sino Biological, and with their proteins in our technology, we could print very nice arrays. basically it's a triplicate. So you have 3 blocks here of 120 different spots, basically 90 mutants and 30 controls, you could say. And yeah, that's the arrays. Here it's stained with an anti-S protein, and the data you see now are basically fluorescent data mainly. But to give you a glimpse how it looks like with a label-free measurement, I myself was unfortunately hit with corona, but I had a blood serum banked already 3 months before that, and you see I had no antibodies at all against COVID-19. And after that, I can see very nicely how protected I'm against the different mutants. And on the left side, you see a therapeutic antibody, which was well known in press because it was used in a very famous and infamous president. And we could always say we tested Tubidus before he himself tested it. And we saw at that time there are gaps. You see here, there are some protections after an infection where this antibody doesn't work. And around Christmas is what also announced by Regeneron that against these mutants, it won't work. And we found that 484 as well as 485 and 486 mutation already a month earlier. But we are too small, and we didn't have a big enough database to really go to the press and say, hey, it won't work at all. But you see here, it already has a predictive power, which was basically proven by reality. And now let's have a look into the larger arrays. So if you have a non-binding antibody, for example, it looks like this. You see here a lot of red staining, which means basically ACE2 is binding, there's no protection, as the antibody is not binding at all. And the SARS-CoV-1 is basically a control for the binding for us. And if you have now a staining with commercial antibodies, for example, this is a S2 antibody, we see very nicely that it binds also the S2 subunits on the array here, for example. And if we have an N protein antibody, you see also it binds the N protein of SARS-CoV-1. You see already here cross-reactivities and can deduce from the kinetics also the binding constants and the binding rates. So this is really a broadband analytical platform with these arrays. And always remember, this is only 1/3 of the whole array. So it's a lot of more data which you have in backend. And you have to each data point here, to each image, you have a complete binding curve of all the steps of all the bindings. If we have a neutralizing antibody, basically one would hope everything is green except the controls. And you see here a very red dot. This is one mutation where this therapeutic antibody is not working at all. So we reproduced it several times, and also our clinician partners tested now if they can reproduce it also in cells for their neutralization assays. And if you have a diagnostic antibodies, because you can decide either to go to the N protein or to the S protein. And as the S protein has a lot of mutations, you see here it fails in 2 cases. So basically, if it's a lateral flow assay, it won't work with these mutants. And this is also quite important if you think that daily testing may be a control measurement to conceal the pandemics at the end. So let's have a look a little bit more into the infection. So as we have all the other coronavirus on the surface, you can directly read out the pre-infection of the persons. You can see this person here, for example, was not so much in contact with different other coronaviruses, whilst this person was. And astonishingly, we see quite often if the persons have a lot of contact with different other coronavirus, they have still no S protein protection, but they have some cross-reactive antibodies with the N protein of SARS-CoV-1 and SARS-CoV-2. And at the moment, it is speculated if such persons have a more severe progression of the disease, because it may be that the immune system simply tracks down first the N proteins and doesn't build up so easily the protective S proteins. But this is pure speculation. Once again, same person here and there, and you see that the N protein antibody amount is tremendously increased, and that this person didn't develop protective antibodies against the E484 mutants, and seemingly would have had a Wuhan infection, because also the antibodies against the D614G was not so high for this person. So you can see here already which mutants may emerge if you have already the panel ready. So this means you're not protected after an infection, or not so good protected here for this ACE2 interaction. Other parts of the T cell system may help you, but here you would say, okay, this may be risky mutants for these persons. And if you use a pool of infected persons, you still see that 484 is weak. This means that it may be an inherent weakness of all persons who have been infected, and this may mean that this mutant will always come up, as it has a higher chance to evade the immune system and still could invade the cells, even if the other mutants are already protective. And as said before, we can also have a look for the vaccination with these arrays. Again, a person quite young, 3 weeks after the first shot, also 484 and some other mutants are not the best, but still good here at that point. And elderly persons, you see quite easily their immune system is not so fast, and still they have great gaps for the immunity. And if you look to a different vaccine, you see the pattern looks similar but not identical. You see some parts or some mutants may be lesser protected, some others may be better protected. Also here in this area you see differences, and here you can basically compare in this pseudo-neutralization assay how do the vaccines develop, but this also has a personalized status also. And you see also quite nicely, none of this person has an N protein cross-reactivity. And the person A, for example, here, 3 weeks after the second shot, has nearly, seemingly for this part, a perfect setup. But also, it seems that this person had an other infection with a different coronavirus whilst the time, because there are antibodies now here. And so let's summarize this COVID-19 arrays. Basically, we have now a nice tool for screening, where we can screen the antibodies against therapy and diagnostics. We can track basically the infection immunity over time against the different mutants. So you see against which mutants are you directly after the infection you are vulnerable. And the same could be done also for vaccines, so that we can have a look for the efficacy, how many antibodies are produced, how protective are they. So in our findings, you have roughly 50-fold amount of antibodies, but the amount of protective antibodies is only 5-fold. So your body produces mass of antibody against the S protein, but is not very specific to go into protectivity, whilst for infection you need to go into protectivity. And this may assist also phase 1 to 3 developments at the moment. It may also help for monitoring phase 4 to have a look, maybe a combination of, let's say, Pfizer and AstraZeneca maybe, or J&J may be helpful, or some other whole virus setups. And you can also compare vaccine upgrades. So if you switch to different mutants, they may also protect better against other upcoming mutants that also can be tested, even in a preclinical phase, because if you think of animals which have been vaccinated, you already see here also if the neutralization works or not. And I simply like to announce here, if you have some analytics, come to us. Also, if you're interested in cooperation, and also if you are interested in these areas, because as far as I heard, no one is offering these areas at the moment. And if enough people come to us, we may sell some of these in a larger batch. And some remarks and conclusion at the end. Where would we position BioCopy, and how would we go into the future? basically, our idea is not only to stay at the mutants which are existing already at the moment, but to make some kind of display or library, phage display or ribosome display. Pen basically sort all those out which don't bind to ACE2. So we know every mutant which is potentially binding and therefore potentially infectious, and use our copier at the end to make a BioCopy S protein array with several 10,000s of proteins, and then basically use the serum of vaccinated persons and flush over these arrays after that in the same way like before now, the ACE2, and then we find basically mutants which have been either addressed or not addressed at all from the immune system after the infection, but still bind ACE2. And these mutants, which are not existing yet, have the dangerous ability to be binders at the end, and therefore they may be evasion mutants and may come up not today, but in the next months or years. And this may be a way that you can directly deduce from the measurement which are the vaccines of tomorrow, so to say. So we can basically identify and defend against these parts before they even start to exist. And this may be a very powerful weapon in vaccines, and maybe also developed in the future. We filed the patent. This is also the reason why we talk today about that. And we're looking for joint development with partners and interested partners to develop it. So basically, I thank for your attentions. If you have questions, ideas, or applications for our copier or for cooperations, then simply contact us. I thank to Sino Biological, as they provided us many proteins partially in front of time, and also my employees and co-workers here at BioCopy. Without their lots of works and ideas, this couldn't have been reached. And you see also we have very artistic people also here. And if you want to have answers, just contact me, ask now, or write an email to one of us. Thank you.
Thank you very much, Gunther. Say, if anybody does have any questions, we do have a chat feature on the platform. So if you have anything you'd like to ask our panelists, then do please type that there. At the moment, we don't have any questions, so let's give people a few moments to see if they can come through. Okay, well, looks
no
like
questions at all.
At the moment, no, doesn't seem to be. Well, I say if anyone does, and we saw your contact details at the end, but if you perhaps also say, it was Gunther.roth at biocopy.ch. If anyone has any questions, you can contact Gunther, and also I'm sure we'll be able to eventually share this, or not, depending on the permissions we're given. But otherwise, I think, let's say no questions. So thank you very much, Gunther, for presenting. So thank BioCopy as well, and Sino Biological for this session. And yes, if there are any questions, we'll pass them through. Otherwise, thank you very much.
Yeah, I thank you to all also for your attention, and yeah, stay healthy and well. Bye bye.